So that's the basics of the structure of DNA. We've got this nice, tightly bound molecule that I can separate very easily. Those two separated halves can replicate, forming identical daughter molecules because of that rigorous complementary base pairing. The next big question we're going to have to ask ourselves is, well, that's great, I've got DNA, I can copy it faithfully, but what do our cells do with this DNA? And to understand that, we need to think about a two-step process. The first of these steps is called transcription. Transcription is going to take the DNA in your nucleus and make a working copy of it, right? Just like a scribe copies a text, the process of transcription will copy the information in your valuable, important, critical DNA and make a working copy of it in a molecule we call RNA. RNA is very similar to DNA. It also has a sugar-phosphate, sugar-phosphate, sugar-phosphate backbone, all covalently bonded to one another, and hanging off each sugar is a particular base. There are two key differences, however, between RNA and DNA. One of them is that we almost always find RNA as a single-stranded molecule. So notice that I have half a double helix here. That's one critical difference. The second critical difference is that when we're building an RNA molecule, we're going to replace the T's, thymines, with U's, uracils. So we're going to use A's, U's, C's, and G's to make up an RNA molecule, but our complementary base pairing rules will still apply. A will pair with U, and C will pair with G. So we want to make an RNA copy. You and I, being eukaryotes, store our DNA molecules in our nuclei, so this process of transcription will take place there in the nucleus. And we're going to use an enzyme to do this. This enzyme is known as RNA polymerase. It's going to make us a polymer, link together a bunch of molecules to make RNA. So RNA polymerase will find the beginning of the gene whose protein you need right now. That beginning site is called a promoter. It's going to bind to your DNA at that promoter, separate the two strands of that molecule, and use one of your DNA strands as a template to make a complementary RNA molecule. We call this complementary RNA molecule an mRNA, or a messenger RNA. It's going to take the message, the directions from our nuclei out into the cytoplasm where our cells can begin to make protein. That's the critical second step we need to think about, how we're going to make protein from a simple RNA molecule. This is known as translation because we're really switching languages here. DNA and RNA are molecules of the same language. They're both nucleic acids. That's what the NA stands for. But now we're going to proteins. Proteins are totally different. These are long chains of amino acids linked together in a particular order. So we need to figure out how an RNA molecule can tell our ribosomes which amino acids to put in which order to make that particular protein we happen to need right now. So to do this, we essentially need a decoder ring. We need to understand the code that allows RNA to convey directions for making protein. And that code, thankfully, is very well known and pretty simple to work with. What we've learned about RNA is that every 3 bases in an RNA molecule form a codon. A codon codes for one particular amino acid. And let me show you how that works. Here, for example, is a very short piece of RNA that we're imagining has already been transcribed in the nucleus, and my artist here has conveniently sort of broken apart the molecule to show me the codons, the series of 3 bases in my RNA molecule that contain directions for making or attaching a single amino acid. They can do this because of the existence of another RNA molecule called tRNA. tRNA stands for transfer RNA, and you might picture it looking something like this. It's literally just an RNA molecule folded up on itself But in this process of folding, it gets two really critical features. One is that at the one end of this molecule, 3 bases are left exposed. We call this an anticodon. The second critical feature is that there's an amino acid attachment site on the other end of our tRNA, our transfer RNA. So I'm going to show that an enzyme in my cytoplasm has attached an amino acid at this other end for me. So the power of this molecule comes into play when RNA is recognized by a ribosome. I'm going to show my ribosome here if I can juggle all these pieces together. So here's my ribosome out in my cytoplasm. It's found my messenger RNA and bound to it. And notice that in the structure of the ribosome, there are a couple pockets here. These pockets are just the right size and the right shape for a tRNA to bind. So here's a codon, AUG. If I understand complementary base pairing, I can predict the sequence of a molecule that would bind here, right? In DNA, A pairs with T, but this is RNA, so I'm going to think about A pairing with U, A pairing with U again, so I want a U, an A, and a C, right? That would be a complementary sequence. So check this out. Here's my tRNA. It can form hydrogen bonds there. It can temporarily bind to that messenger RNA and bring into position the correct amino acid. When it does this, my ribosome is going to slide down, bring into position another tRNA carrying the next amino acid attached to it. I'll show you this in class because clearly it's not going to work for me right here. But we'll play with this again. But the idea is that my tRNA can bring the correct amino acid to the correct position and then catalyze the formation of a bond between these two. So we're going to use these tRNAs as my magic decoder rings to figure out what sequence of amino acids are required for the particular protein I need right now. So there's a quick overview. We'll be adding some more details, but we essentially want to understand the structure of DNA, especially complementary base pairing, how that allows us to replicate DNA. So you might practice that. If you know a sequence of DNA, can you tell me the other half of that molecule, its complementary strand? We need to understand that, and then we need to understand that two-step process, transcription and translation, that allows us to make a working copy of DNA in our cytoplasm. That's the messenger RNA, which we can translate using ribosomes and tRNAs to make a fully functional protein. So we'll look at each of those steps in more detail later, but happy reading. That should give us a good start.